Hi, in this tutorial we are going to study the structure of the 20 amino acids. Amino acids have two functional groups. They have the amine functional group and they have the carboxylic acid functional group. All amino acids have a hydrogen atom for the third bond, and the fourth bond to the alpha carbon is the R group, and the R group is different between the 20 amino acids. Based on the identity of the R group, so based on the polarity and the charge on the R group, amino acids can be divided into different groups. So we are going to start from the non-polar or hydrophobic amino acids that have linear or aliphatic side chains. The first one in this group is alanine. I also included the 3-letter and the 1-letter code for each amino acid. So the side chain for alanine is a methyl group. Next is valine. The side chain for valine is an isopropyl group, as shown here. So isopropyl is a chain of 3 carbons, 1, 2, 3, and the second carbon is bonded to the alpha carbon. Next is isoleucine. The side chain for isoleucine is a sec-butyl group. So sec-butyl group is a chain of 4 carbons, 1, 2, 3, 4. So 3 of them are linear, and the fourth one, which is a methyl group, is bonded to the first carbon. Leucine and isoleucine have very similar side chains, as shown here. The only difference is that the methyl group is on the second carbon instead of on the first carbon, so the side chain for leucine is an isobutyl group. And the last one in the linear side chains is methionine. The side chain for methionine is as shown here. So 2 CH2 groups, 1, 2, bonded to a sulfur, and this sulfur is bonded to a methyl group. Now we are going to look at the amino acids with aromatic side chains. The first one in this group is phenylalanine. The side chain for phenylalanine is as shown here. So it is basically an alanine bonded to a phenyl group. So this is a phenyl or benzene ring. And the last one in the aromatic side chain group is tryptophan. The side chain for tryptophan is as shown here. So it is a CH2 bonded to an indole functional group. And now we are going to look at polar amino acids with neutral or uncharged side chains. So the first one in this group is serine. The side chain for serine is as shown here. So it has a CH2 bonded to an OH functional group. Threonine is very similar to serine. The only difference is that in threonine it is CH3 instead of a hydrogen atom. Next is asparagine. The side chain for asparagine is CH2 bonded to an amide functional group. Glutamine and asparagine are very similar because they both have the amide functional group. The only difference is that glutamine has one more CH2. And the last one in the polar amino acids with neutral side chains is tyrosine. The side chain for tyrosine is as shown here. So tyrosine is very similar to phenylalanine, but it has a hydroxyl group, and therefore it is polar. Now we are going to look at polar amino acids with charged side chains. We are going to look at the negatively charged amino acids first. There are 2 of them. The first one is aspartic acid. The side chain for aspartic acid is as shown here. So it is a CH2 bonded to a carboxylic acid functional group. Glutamic acid is very similar to aspartic acid because they both have the carboxylic acid functional group, but glutamic acid has one more CH2. Now we are going to look at the positively charged amino acids, and we are going to start from arginine. The side chain for arginine is as shown here. So it has 3 carbon chains, 1, 2, 3, bonded to a guanidine functional group. Next is histidine. The side chain for histidine is as shown here, so it has a CH2 bonded to an imidazole functional group. And the last one in the positively charged amino acids group is lysine. The side chain for lysine is simple compared to histidine or arginine. So it is 4 carbon chains, 1, 2, 3, 4, bonded to an amine functional group. And now we're going to look at 3 amino acids that have different characteristics. The first one is cysteine, and the R group for cysteine is the CH2 bonded to an ACH. And cysteine is a polar molecule, but it cannot form hydrogen bonding interaction. Instead, it can form a disulfide bond with another cysteine molecule. Next is glycine. Glycine is the simplest amino acid because the R group for glycine is a hydrogen atom. And since glycine has 2 hydrogen atoms, it is a chiral molecule, and glycine is a nonpolar. And the last one is proline. Proline is very different from all of the amino acids because the R group for proline, which is 3 carbon chains, 1, 2, 3, is bonded to the backbone amine as shown here. And proline is a nonpolar amino acid. So now I'm going to give you guys some tips to help you to quickly draw and learn the structure of the amino acids. As we know, all amino acids have the same backbone. The amine, the carboxylic acid, and the hydrogen atom are the same in all of the amino acids, and therefore you can represent the backbone with an X when you draw. And the only thing you have to draw is the R group. So you have to draw it several times to learn the structure of the 20 amino acids. So for example, if we want to draw the structure of arginine, you can represent the backbone with an X as shown here, and you only have to draw the R group, which is 3 carbon chains, 1, 2, 3, bonded to a guanidine functional group. If you want to draw tryptophan, you represent the backbone with an X as shown here, and you only draw the R group, which is the CH2 bonded to an indole functional group. And if you want to draw leucine, you represent the backbone with an X, and you only draw the R group, which is 4 carbon chains, 1, 2, 3, and then the 4th one is bonded to the 2nd carbon. So this will help you learn the structure of the 20 amino acids faster. And that's it for this tutorial. Thank you for watching, and please let me know if you want me to do more videos in biochemistry. I will do my best to address your questions, and thank you. See you in the next video.